Good afternoon and welcome to Tabula Rasa. We are an online education platform where we try and cater to students who do not have as much access to the current ongoing global wave of online classes, and we also cater to students who want to study and want to understand their subjects better than the average education institutes could provide them. Since education is our primary motto and education for everyone is our objective, we have also come up with this initiative where we will be discussing various different issues and various different academic as well as non-academic topics live online via live feeds in the form of live webinars. Thank you for joining us here today. Today we have with us Dr. Sumit Mukherjee. Dr. Mukherjee is a postdoctoral researcher and Israeli Faculty of Medicine at the Bar Ilan University, Israel. He will be talking on coronavirus and a study in immunity. There have been various questions raised since the coronavirus pandemic and its implications became popular globally. Various questions initially ranged from what the virus is, how it infects people, and what to do about it. Today these questions have been more or less answered, and things have changed from flattening the curves to more or less down to what are we going to do now? What is the solution to this? What is the possible cure? When are we going to get a vaccine? Dr. Mukherjee will be talking with us today on what makes a vaccine, what it takes to make a vaccine for a condition, for a disease as global and as difficult to treat, as difficult to understand as the coronavirus, the COVID-19. Dr. Mukherjee, I'll transfer right away to you.
First of all, thank you for inviting me, Tabula Rasa, and good afternoon all. Just a minute, I'm just sharing my screen. Am I audible? Hello?
Yes sir, you are audible.
Hello?
Yes sir, you are audible. You are quite audible and visible
Yes.
as well.
Yes.
Please go ahead.
Okay, just a minute. So today I will discuss the present status of the coronavirus vaccine and why it takes so long. So before explaining the vaccine, I want to talk about a little bit about the immunity or the body immunity. So what is immunity? So immunity is the ability of an organism to resist disease through preventing the development of a pathogenic microorganism. So basically, immunity can be classified into two types: innate immunity and the adaptive immunity. So innate immunity activates the fast response from our body to a pathogen, and the skin itself a part of the innate immunity. It is the primary defense of the body that stops harmful pathogens or the organisms from entering the body. So however, pathogen can still break into the body by means of air, food or water. So the second defense of the body is the form of mucus, which is attached the pathogen to itself and kills it right there. It is destroyed by the complement system, which involves a set of proteins. So now let us assume a pathogen is even more powerful and it escapes from skin and the mucus as well. Then it further goes inside and confronts the cells called phagocytosis and the natural killer cells. All these cells detect the pathogen and attack and destroy it right there by a process called phagocytosis. So this is basically the innate immune mechanism. It is the fast and immediate response of our body when a particular pathogen tries trying to enter in our body. Then if the pathogen still survives, okay, and the phagocytosis and the NK cell, then our body activates the secondary immunity, which is called the adaptive immunity. Okay, the adaptive immune system evolved early vertebrates and allow for a stronger immune response as well as immunological memory, where each pathogen is remembered by a signature antigen. The cell of the adaptive immune system are specifically types of leukocyte, is called the lymphocytes. Is B cell and T cells are the major two types of lymphocyte and are derived from the hematopoietic stem cells in the bone marrow. And B cell is involved in the humoral immune response, whereas T cells are involved in the cell mediated immune response. So basically, based on this, we can classify the immunity into two types: is humoral immunity as well as the cell mediated immunity. So generally, humoral immunity refers to the antibody mediated response. In this humoral immunity, the antibodies are produced by the plasma cells and protect the host from infection in three main ways. First, by binding to pathogens to inhibit their toxic effects or by the neutralizations. Second, by coating pathogens and facilitating their uptake and killing by phagocytosis or opsonization. And third, by activating the complement cascade in our immune systems. And the cell mediated immunity is the immunity is mediated by the antigen specific T cells. Okay, so now I am coming to explain the more details in humoral and cell mediated immunity. As I say, that humoral immunity is mainly the antibody mediated response, and the antibody is produced by the plasma cell and protect the host from the infections by binding the pathogens, and it is generally involved in the phagocytosis process. And in case of cellular immunity, the T cells are it is based on the antigen specific T cells. So we know that the T cells are produced in the bone marrow and are matured in the thymus, and after they enter the bloodstream, T cells occur can found in blood as well as in the lymphoid tissue. The antigens should be presented on the surface of the antigen presenting cells along with the major histocompatibility complex, or the MHC we called. Once the T cells encounter an antigen, they proliferate and differentiate into armed effector cells. The cytotoxic T cells, as we can see that there is in CTL, is called the cytotoxic T cell, which can destroy the infected cells by inducing apoptosis. And the T helper cells, or the we can call the helper T cells. Generally, it can stimulate the plasma B cells to produce the antibodies. Okay, so when basically B cell and T cells are activated and begin to replicate, some of their offspring become long-lived memory cells. Throughout the lifetime of human, these memory cells remember each specific pathogen encountered and can mount a strong response if the pathogen is detected again. This is adaptive because it occurs during the lifetime of an individual as an adaptation to infection with that pathogen and prepares the immune response for future challenges. So, immunological memory can be in the form of either passive short term memory or active long term memory. So, we can generate the adaptive immune response by vaccinations. So how the vaccination can stimulate the adaptive immune response? So it is an important things today's talk we will address. So vaccination is an effective way to improve the public health by building up adaptive immunity to a target pathogen. It's maybe virus, bacteria, fungal, or any kind of pathogenic organisms. So a vaccine works by training the immune system to recognize and combat pathogens, either viruses, bacteria, fungi, or it can be a protozoa. So to do this, a certain molecules from the pathogen must be introduced into the body to trigger an immune response. These molecules are called antigen, and they are present on all of the pathogens. By injecting these antigens into the body, the immune system can safely learn to recognize them as invaders, produce antibodies, and remember them for the future. So then, if the pathogen again reappears, the immune system will recognize the antigen immediately and attack aggressively well before the pathogen can spread and cause a disease. So, as I say that vaccination is the program from by which vaccine is introduced into the population, and immunization programs and the development of a new vaccines play an important role in protecting individual against a certain diseases. And vaccination works by safely exposing individuals to a specific pathogenic microbes, artificially increasing their immunity to it. So, what is the main importance of the vaccinations? So, vaccine don't just work on an individual level; they protect the entire populations. Once enough people are immunized, opportunities for an outbreak of disease become so low. Even people who are not immunized benefit. Essentially, a pathogen a pathogen or the pathogenic virus simply won't have enough eligible hosts to establish a foothold and will eventually die out entirely. This phenomenon is called the herd immunity or the community immunity, and it has allowed one devastating the disease to be eliminated entirely without needing a vaccine every individual. So now we are coming to the types of vaccinations. So there are several kind of vaccinations are there. The main two are the live attenuated vaccine or the inactivated vaccine. So in case of live attenuated vaccine, this is the weakened version of the actual virus or the pathogens. So the advantage of using this vaccine is can stimulate a robust immune response without causing serious disease. But the disadvantage is it may not be safe for those with immunocompromised. Second, the inactivated vaccine. Inactivated vaccine is a kind of vaccine that use the whole virus or the pathogen after it has been killed with the heat or by chemicals. It is safe because the pathogen is already dead, but the main disadvantage is it is not effective as live virus or the pathogen. Some previous inactivated vaccine have made the disease worse. So then generally people are also trying to develop the inactivated vaccines for the coronavirus. I will discuss in the later slide. And the one of the most popular inactivated vaccine is the polio vaccines. Now we can another kind of vaccine is there is called the DNA or the RNA vaccines, where the directly the molecules direct molecules like DNA or RNA can be used uh, from the pathogens to develop the immune response against a certain viral against a particular uh, immune response. So the main advantage is it is very easy and quick to design, because just we can isolate the DNA or the RNA and it can introduce into the host to generate the immune response. But the disadvantage, because never have been done before, there are no licensed DNA and RNA vaccines currently in use. Next, I am coming to the subunit vaccines. So subunit vaccines are also called generally a peptide vaccines. These vaccines can use a piece of the virus, mainly from the part of a viral viral protein sequences. So or it I will discuss it later about the subunit vaccine also. And this peptide or the subunit vaccines having several advantage because it can focus the immune response on the most important part of the pathogens and the protection and cannot cause the infection. But the disadvantage because it may not simulate strong response and other chemicals maybe need to added the boost long term immunity. Other chemicals in terms of adjuvants, we can use the adjuvants with this subunit or the peptide vaccine to make it more powerful. So there are several peptide based vaccine are available. It's called the pertussis, hepatitis C vaccine, HPV vaccines. Next, and the last is the viral vector vaccines. What is the viral vector vaccines? This approach takes a harmless viral virus and is used it to deliver viral genes to build an immunity. So the main advantage of this viral vector vaccine is it is a live virus tend to generate the stronger immune response than the dead virus or the subunit vaccine. But the disadvantage is it is important to pick a viral vector that is truly safe, and immune response to the viral vector could make the vaccine less effective. So generally, the now in market Ebola vaccines are present, which is generated based on this viral vector. And at present, as we all of us know that the University of Oxford and AstraZeneca are they are collaborating and produces a viral vector based vaccine for COVID-19, which is under clinical trials. So now question is that why it takes too much time to develop a vaccines. So these figures are generally represent the timeline for the vaccine development. So the general stage of the development cycle of the vaccines are several phases or the stage. The first stage is the exploratory stage. In the exploratory stage, the researcher or the academicians are generally working on to identify the vaccine targets from the pathogens, and they are testing it that if this vaccine target is efficient or not, or if these vaccines targets can be introduced into the animals and it can generate the immune response or not. Then if it some vaccine candidates are detected using the basic research in the exploratory stage, then it can go to the preclinical testing. In the preclinical testing is the generally it is the initial safety testing is usually carried out in animals to give an idea of responses in humans. These are also used to see how effective the vaccine at preventing the disease and allows the scientists and the researchers to adapt the vaccine. So during an outbreak, different research groups often work together to speed up this process. Generally, this preclinical testing can take 5 to 10 years. Then, if a vaccine is passed through the preclinical testing, then it can goes into the clinical testing phase. So the clinical testing phase into three types or the three processes. During phase 1, a small group of people receive the trial vaccine. In phase 2, the clinical study is expanded and the vaccine is given to the people who have characteristics similar to those for whom the new vaccine is intended. So in case of phase 2, near about 100 or 200 uh, volunteers, the vaccine can be given to the 100 or 200 volunteers. And if the phase 1 and phase 2 clinical trials are passed, then it can go to the phase 3 trials. In phase 3 trials, the vaccine is given to more than 1,000 of people and tested for efficacy and safety. So generally, in clinical testing could take near about 5 to 8 years, because after the phase 1, when the small group of people have the vaccinations, then their immune response as well as their if they have some kind of side effects, it can be evaluated just after the 1 or 2 years after the vaccinations. Then it can goes to the phase 2. Then similarly, after it's successful, if it's successful in the phase 2, then it can goes to the phase 3. So generally it took to the 5 to 8 years for the clinical testings. But in case of SARS-CoV-2 vaccinations, as this is a more it is a global pandemic, so we need the vaccine very urgent. So then this phase 1 and 2 trials are combined together, and most of many vaccines are under developing for this phase 1 and phase 2. And now the Oxford vaccine is it goes to the phase 3 trials also. So after the phase 3 trials are succeed, then it can go to the vaccine can generally, when it is passed after the phase 3 trials, then it can be produced at a small scale, and then it can be used for further clinical trials or the phase 4 trials. However, the significant manufacturing capacity, such an infrastructure, personal as well as an equipment, will be needed to produce larger quantities of a vaccine for use. So, but the quality control step is also very important during this vaccine production in the industry, and all of these processes are very carefully monitored. So after it passed the phase 4 monitoring, then it can get license. And once it's licensed, policy must be developed to decide how to prioritize, um, how to give it based on the priority to those who should be vaccinated, such as those in the most high risk group of populations, and the exact the geographical locations, such as the Ebola vaccines are mainly given to the populations of the people from the Africa. So along the main we can say that in this way, if any of these vaccine candidates are shown to be unsafe or inactivate in also ineffective, not inactivate, ineffective, then researcher must return to the lab to develop a new candidates. So now we can understand this why this is the reason why the vaccine development can be long and it is an uncertain process. So at present, only 10 to 12% vaccine can be successful for after the clinical trials. So this is an pictorial representations of the vaccine timeline of the vaccines. So we can see that in case of polio vaccines, the first epidemic in the USA was 1894, but it can take near about 60 years to develop the polio vaccines just after the outbreak. Similarly, in case of Ebola, the first Ebola genome was sequenced in 1993. Then we all know about the largest outbreak of West Africa in 2014, Ebola outbreak. The Canadian team first published the Ebola vaccine in 2005, and the first clinical trials was in 2015 for the Ebola vaccines, and the vaccines approved in 2019. So generally, it took near about 14 to 15 years to develop the Ebola vaccines. Similarly, in SARS-CoV-1, the first coronavirus outbreak is SARS-CoV-1. It was in during this 2002 and 3. Then the SARS genome sequenced in 2003, but first clinical trial was in 2005. But at there is near about 17 years no vaccine. Then SARS-CoV-1 is mutated and generated new strain is called the Mars outbreak, which appeared during this 2012 and 13 in Saudi Arabia. Then their scientists are trying to develop a vaccine against the Mars in 2016. It is also in the first clinical trial, but still there is no vaccines. And just after the 8 years from the Mars outbreak in 2019 December, we all know that there is a global pandemic caused by the coronavirus is based on SARS-CoV-2, and there are several vaccines. More than 30 vaccines are under research and 14 of them are past clinical trials, fast clinical trials, and they are under process. So we can expect that the SARS-CoV-2 vaccine will be available in the next 12 to 18 months, but we don't know if it is capable to generate the sufficient immune response in human in the large scale, and as well as it does not have any side effect. So this is the projected timeline for COVID-19 vaccine trials. So there are 14 COVID vaccines which are showing in these pictures, which are under clinical trials. There are two DNA vaccines, three RNA vaccines, four protein based vaccines, three viral vector based vaccines, and two inactive vaccines. Just a minute. So now we are discussing about the Oxford COVID-19 Oxford vaccine trial. So this is the most important vaccines because it passes the phase 2 clinical trials, and now it is under the phase 3 clinical trials. So this COVID-19 Oxford vaccine is also called the AZD 1222. It is developed by the Oxford University. It is an investigational vaccine against SARS-CoV-2 in the development for the prevention of COVID-19. This works by using a viral vector. It's mainly based on the chimpanzee adenovirus. Oxford 1 is named, the name of the vector is CHADOX1, is the chimpanzee adenovirus Oxford 1. It is based on a weakened version of common cold adenovirus that contains the genetic material of the SARS-CoV-2 spike protein. So the chimpanzee adenovirus is modified, that which is unable to cause disease. Then the SARS-CoV-2 protein, spike proteins, part of the spike protein, are introduced in this viral vector, and it can generate a new vaccine candidate. And after the vaccination of the surface spike protein is produced, it can priming the immune response to attack the virus if it is later infects the body, and it protecting it against the COVID-19. So the University of Oxford is working with the UK based famous global biopharmaceutical company AstraZeneca for the further development of its large scale manufacture and the potential distribution of these vaccines. The result of the phase 1 and 2 clinical trials of this vaccine is published recently in the scientific journal Lancet, and it indicates there is no early safety concern, and it induces strong immune response in both part of the immune systems. And this vaccine provoked a T cell response within 14 days of vaccination and an antibody response within 28 days. Okay, so now I am discussing uh, recent work from our lab in the Berlin University of Israel, and we are also trying to develop a vaccine which is based on the peptide-based vaccines for SARS-CoV-2. The initial study for vaccine designing is recently published in the journal Vaccines. So first, we should know that what is peptide vaccines and epitopes. So peptide vaccines are an attractive strategy that relies on usage of short peptide fragments of pathogens to induction of highly targeted immune responses, consequently avoiding allergenic sequences. So in case of peptide vaccines, a small part of the viral or the pathogen proteins are generally considered for designing a vaccine. So this small part which can induce the immune response is called the epitope. So epitope is typically a pathogen protein segment that can trigger a cellular immune response mediated by T or the B cells. And combination of multiple epitopes are useful for development of effective peptide vaccines. So the main approach is to from the pathogen to identify the target or the protein then identify the epitope, then then this combination of this epitope, we can generate the multi epitope vaccine, is called the peptide vaccines, and then these peptide vaccines can use for the vaccination. So the first and foremost thing we have to consider to designing a peptide vaccines um, is for to identify the immunodominant epitopes. So what is the immunodominant epitopes? Immunodominant epitopes that can induce protective immune response in terms of both humoral immunity as well as cell mediated immunity against a desired pathogens. So immunodominant epitopes are the part from the pathogens protein which should induce both humoral as well as cell mediated immunity, and it should have been capabilities of generating protective immune responses in human. So in this study, we use a reverse vaccinology based approach. So in these modern generations, when the vast amount of pathogen genome sequences are published, because we all know that there is a next generation sequencing technologies can generate the can sequence the genome and generate the vast amount of sequence data very rapid. So um, the with the advent of this next generation sequencing technology, along with the bioinformatics or the computational advancement of the computational algorithm, it is very interesting to study the pathogen genome sequence using the various bioinformatics tools. So if we have a particular pathogen genome sequences, then we can identify the important antigenic part from their pathogenic protein sequences using various computational algorithms. And then if once we can identify if this part at the could be an effective antigens, then we can go for the further experimental studies and clinical trials. So this is a typical representations where a particular um, vaccine candidate is screening using the computational algorithm from a pathogens called T parva. So first, the researcher took the genome sequences of the T parva, then using the in silico antigen predictions, that means using several computational algorithms and softwares, they predicted which part could be an effective antigens from their protein sequences. Then they characterized in vitro of this predicted antigen and selected which could be the immunodominant peptides or the epitopes, and it can be then then these epitopes could be introduced into the host. Here is the catalyst. So we know that if we considering the genome sequences of the SARS-CoV-2, so there are mainly SARS-CoV-2 consists on mainly 5 primary proteins. So the most important one is the spike glycoprotein, by which the virus can get into the cell. The M protein, which is important for the virus germination and the envelope formation, and the nucleocapsid protein, the N protein, which is related to the formation of nucleocapsid in the RNA genome. And also there is an E protein or the envelope protein. It is the smallest, and most of the function is the mysterious of this protein. So how we identify the immunodominant epitopes from the SARS-CoV-2 genome? So first we took all the available sequence of SARS-CoV-2 genome sequences from the various databases, and then we identified the possible B cell and T cell epitopes using various tools. As we can see that we have identified the B cell epitopes using the most popular tools called LBTOF, ABCPRED, and BAPIPRED, and we identified the T cell epitopes using TEPI tools, NetMHCPAN, NLHPRED, CTLPRED. And once we identified all the B cell and T cell epitopes from on the SARS-CoV-2, then we mapped if those B cell and T cell epitopes are also present in the SARS-CoV protein or not. So for this reason, we downloaded all the SARS-CoV experimentally validated epitope sequences. Then we mapped the immunogenic regions based on the B cell and T cell epitopes, and we also predicted their protective antigen ability using Vaxgen, and we selected the epitopes which are 100% identical with experimentally validated epitopes of SARS-CoV-2. So that means we selected the regions from proteins of the SARS-CoV-2 where we can map both B cell as well as T cell epitopes. So that means this region can introduce both humoral as well as cell mediated immunity. So for once we identified the possible immunodominant epitopes, then we are further analyzing few things to validate if those epitopes could be useful for the vaccine target or not. So the first we analyzed their mutation. So we took all the sequenced genomes from the different geographic locations of the SARS-CoV-2. So that means from the SARS-CoV-2 isolates from China, USA, India, Brazil, and Europe, different places. Then we perform the mutation analysis and check if these epitopes could mutated or not. And we found that among few epitopes are mutated, but most of them are not. So we selected those epitopes which are not mutated. Then we perform the population coverage analysis. So what is population coverage analysis? Population coverage analysis is mainly based on the mainly based on the analysis, which is if a vaccine can produce using the epitopes, then if it can cover almost all the global population or not. So this is generally say that if we design a vaccine using this predicted epitopes, if this vaccine can cover almost all the global population or it can cover a certain group of populations. So then further we analyze their structural and molecular docking analysis to understand if it is exactly bind with the human system, immune system or not. And further we also perform the analysis of their predictions of antigenicity, toxicity, such different things. And we found that from the SARS-CoV-2 proteome, there are 25 epitopes in the from 15 different regions of three SARS-CoV-2 proteins: two from the membrane glycoprotein, five from the nucleocapsid protein, and eight from the spike glycoprotein, which can generate both humoral as well as cell-mediated immunity. But based on this global population coverage, we found that only seven among the 25, only seven of the epitopes can generate more than 90%, can cover more than 90% global populations, and as well as these seven epitopes are can bind with the human major histocompatibility complex in the immune system with high affinity, and there is no issue with the toxicity or the allergenicity. So here we map the epitopes in the protein structure of the SARS-CoV-2. In the left side, in the upper portions, A image represent the nucleocapsid phosphoprotein, where the highlighted part represents the potential epitope that we identified as a vaccine candidate. In the second, in the B membrane glycoprotein, there is two epitope, and in the spike protein we have identified 8 epitopes. So this is an just molecular docking experiment. We have found that with this MSC human MSC class 2, this green color represent the human immune protein like the MSC class 2 protein, and the orange thing is represent the mainly viral epitopes is from the spike protein, and we can see there is a high affinity binding with the human MSC2. So what is the main key findings of our study? In the present study, we identified immunodominant epitopes from SARS-CoV-2 proteins that could induce both humoral as well as cell-mediated immune response in humans, and the 7 potential immunodominant epitopes that were shortlisted based on their higher conservancy, higher global population coverage, and the significant interaction to MSC class 1 and class 2 alleles with high affinity. And these epitopes have a low risk of being allergen, toxic, or generating any kind of autoimmune reactions. So now our collaborator will start the preclinical trials uh, in the Berlin University based on the findings, and also we are looking for the industry partner for the clinical trials of this immunodominant epitopes as in vaccine. So these are the few news coverage of our findings based on our findings and the ongoing work is in Times of Israel, Times of India, and the Jerusalem Post. And thank you all for your attention. And again, thank you, Tabula Rasa, for inviting me for discussing the vaccines, which is most important thing right now. And people are looking for when the vaccine will be available. And as a researcher, we can say that it will take generally if we want to get a effective vaccines with a very minimal chance of any kind of side effect, then we should wait for near about 1 year, then we can get a effective vaccine against the SARS-CoV-2. Thank you.
Thank you, Dr. Mukherjee, for that very illuminating lecture. And I'm certain that with your lecture you have been able to clarify a lot of the doubts that people have had today, are having today, regarding the development, regarding the vaccination process, regarding how immunity develops and everything. So from our audiences, we have a few questions for you, sir.
Okay.
So the first question that we have is, you're saying that these vaccines, these diseases, when they infect a large section of the population, one of the ways to actually protect the population is herd immunity. So the question that we have is how feasible it is, reaching a herd immunity for a country like India. How feasible and how actually possible it is, for considering the population, considering the population density. How many people, in very crude terms, how many people will have to die to reach herd immunity?
Yes, so herd immunity, also called, as I say, that it is also called the community immunity. So when suppose vaccination, particular vaccination could be effective for generating the herd immunity in this community, and similar in case of India, so it can generally take near about few years. If a vaccination program is start, then first we cannot give the vaccination to all the populations. So generally we have to select which people are in at the high risk to generate to develop this disease, particular disease such as COVID-19. So first we need to give the vaccination of this community, and then after that we can give the vaccination to the entire population. And generally we can say that it can take near about 2 to 3 years to generate a herd immunity or the community immunity against a certain diseases. So as you can see, for the cases of polio, it can take near about 10 to 15 years to generate an herd immunity. At this present time, there is very few or no cases in for polio in India. But at a time if you are going to the past, near about 20-25 years ago, there is some people are there who have an high risk. They have a kind of polio, and and we are going before the 50 years, there are lots of people have the polio. So as we can generate the herd immunity, so there is a chance that these particular pathogens can eliminate from the a certain community of the populations from a country or across the globe.
Okay, so you're saying basically is the herd immunity concept is feasible, but only applicable to a population with a smaller country with a smaller population or a smaller population density size.
Exactly. Exactly. It will take for a larger population like India and China, it will take near about few years to generate the herd immunity. And we have to also look for that if we develop a vaccine against a certain pathogen, we can use the certain part of the pathogen which are not evolving. That means in case of pathogens, especially for the virus are rapidly evolving. So if a particular pathogen, we develop a vaccine based on the part of the pathogens, but if the pathogen or the genomic sequence is evolving such a way that this particular part which we used in the vaccine are changed, so then there is a chance that this pathogen can escape the immune system. So it is also mutations, and we have to predict which part are less evolving in the pathogen sequences and which part are the faster evolving in the pathogen sequences. So so designing the vaccine for a particular the effective vaccines, so we have to consider the part which are not rapidly evolving. Okay, so for generating the herd immunity, we have to consider the part which are less mutated or not rapidly evolving, and also we have to for a larger population, it can take near about 3 to 4 years for developing a community immunity. That's it.
Right. So basically, immunity process for a country like India or China should be the vaccination process should include parts of herd immunity and parts of the designed structured vaccination.
Yes.
Okay. Okay.
Can you repeat? Is there a question or anything?
No, this is just an observation, sir.
Okay. Observation.
So
Okay, thank you.
So the next question that we have is, say you're saying that the vaccination process development needs time. It goes through a number of legal channels. It goes through a number of approval processes. So isn't it the prerogative of institutes like the drug regulation institutes basically? So don't they have any authority to regulate or amend laws necessary, which will speed up the process to get the vaccine developed, speed up the process of human trials and everything, and or get them from other states if they have progressed further? Is there any?
So yes, this is a very interesting question. So for the vaccine development, there are, as I say, that there is lots of stage. So in academics and the researcher or the scientist, they are mainly involved in the up to the preclinical trials based on the animal testing. But when it goes to the clinical testing, then the academicians or the researchers need an industry partner. Otherwise, they can produce a large amount of vaccines and they can give to the populations. So when a particular vaccine is developed and it is undergoing to the phase 2 or the phase 3 clinical trials, then there is lots of safety and regulation issue. And it is mainly this safety and regulation issue is mainly varies from the countries to countries, and although the WHO is controlled everything, but still there is regulations from India. It's also different from the Israel or any European country. So generally it could take long time to get the approvals. So generally you can see that in case of SARS-CoV-2, as it is a global pandemic, so people, this approval are getting fast. But in case of other normal epidemics, not the pandemics, it can take near about 5 to 6 years to get the approvals, clinical trials, and also one of the most important things for the clinical trials, when the clinical trial results has come, then near about we have to wait for near about 6 months to 1 year to check if there is any kind of side effect, or if the vaccine can generate the long term immunity, or it can generate the short term immunity. So based on this, then the authorities are sanctioned the license for a particular vaccines. Okay.
Right. So, is there any way, like, if any common person comes and goes up to the administration or goes up to scientists and asks them, "Sir, we need the vaccine right now. We don't have time."
Yes.
So, what then would be the answer? Why is it taking
There
so
is
long?
no answer, because we, as a scientist or the researcher, we can say that yes, a particular, if we develop a vaccines, we if we identified a vaccine target and we can test it in the animal models, yes, it is succeed, and yeah, if it can generally took near about 6 to 8 months to test if the vaccine target is effective or not. But we don't know if it is working in all of the human from a particular population or across the globe or not. So then we should go for the clinical trials, and this clinical trial is mainly in the hand of the government, and as well as is based on the industry partner. So generally, as a scientist or researcher, we are also in the same stage of the peoples who are mainly want to know that this exactly vaccine when it will come. So generally, this clinical trials is the most important phase of the vaccinations program. So it is not in the hand of generally the scientists or the researchers or the academicians.
Okay, that's a pretty dire picture. But anyway, right. So the next question that we have for you, say you as a scientist or as a researcher,
Yes.
involved very closely involved with the development of models, vaccine candidates for this condition. What do you think? You have closely observed the conditions worldwide. You have closely observed how people have been dying, how people are getting affected here and there, and it's a pandemic situation, and it happened in a matter of a few months span. It covered the entire globe. So, what, in your opinion, could be a few steps that the authorities a government should take or could take to
Yes.
prevent this from happening again?
Yes. The first and foremost step it should take that the government should take that they some release more funding in the basic research. Okay. So then this researcher can able to develop or the identify a particular vaccines, because as I say that for identification or development of vaccines only there is a 10 to 12% success rate, and mostly 80 to 90% is the failure rate. So by considering this thing, the researcher also need the money. Okay, so this is the most important thing. Second, for particular this clinical trials and everything for approval, and the authorities should take the government authorities should take the rapid decisions based on the rapid decisions of the approval. And the third important thing is to there is there have to generate the industry as well as the academic partnership to develop the vaccinations. So these three is the most important step to for develop a new vaccine program, and as well as this could be very useful for if there is further any pandemic or any epidemics in the near future. Okay.
Right. So, you're saying that governments, when they spend more money in research, in testing out possibilities of treating people, testing out possible vaccine candidates, these tests become take up a lot of priorities. So far, So far we have come across,
Yes.
so far we have come across various different types of tests for these for these this condition, identifying individuals. We have seen asymptomatic individuals spreading widely. So
Yes.
my question is, how important do you think these tests are? Different sorts of tests are, how important they could be, or they could be connected, whether they could be connected at all in the development process of a vaccine. Are these tests important at all?
In test, what's your question? In the test of for the detection of the coronavirus, or what?
Yeah, exactly. For this particular condition is what I'm asking. Tests have been many. The tests have become very important. Initially, there were different, very, very not very specific tests going on. But now these tests have become very, very specific. So, how important do you think this testing process has been? In the development process of the vaccine.
Yes, yes. As I say that this testing process is for based on this we can for the vaccination. The vaccine at first, if we develop a vaccine, then it should be given to the population who at a high risk. So we can say based on the recent few studies, we can understand that the peoples who are older age with the cardiovascular diseases or the problem or the diabetes, they have a high risk of chances to also have their possibilities to get the COVID-19. And the people who are generally asymptomatic are, you can see that their vitamin D levels, or the most important thing for developing the COVID-19 severity is the cytokine storm, where the in case of any other viral infections, you can see there is a high leukocyte count, but in case of COVID-19 there is a low. Okay, so we have to check exactly in which populations have this signature of the severity. So then we have to give in the vaccine of this particular populations, and then after that we can give into the other populations. So based on this we can and generally develop the vaccination program effectively. Yes.
Right. So at this point, Dr. Mukherjee, let me come back to a bit of your presentation and your particular work in the progress of this vaccine development process. So we have heard a lot of things about this virus. It infects people. Sometimes it has been said that it's not really very clear how it infects people or why it infects people. So what is the most virulent part of the COVID-19 pathogen? Most virulent as in which is the actual virulent part of the pathogen, the coronavirus? And in your particular sequence, you have said that you have picked out peptides from the exterior parts
Different,
of the.
yes.
Yeah, exactly. So how do you select these peptides and convert them into the vaccination candidates? How do you select this portion? Is
Yes, yes, I will discuss it. It's a very interesting question. So we selected the part of the virulent portions from the pathogens of the viral protein based on the computational analysis. So basically, I am a computational biologist. Okay, so or I am specifically work on genomics. So in this case of SARS-CoV-2, first we took all the genome sequences which are available in the databases. Okay, and based on these genome sequences, there are lots of programs which are previously developed based on the machine learning as well as based on the probabilistic modeling. We predicted the potential B cell and T cells, antigens or the epitopes from their genome sequences, or based on the not the genome sequences, based on their protein sequences. So then we predicted if these predicted epitopes can generate the immune response in human or not. There is also different kind of predictive models are there. We use the predictive models to identify if a particular epitopes can generate the immune response of human, or it can be a protective on antigens or not. Then Further, we tested their allergenicity, antigenicity, or autoimmune reactivity. Is all are the bioinformatics predictions. But the one of the most important thing that we considered these predictions and map our predictive epitopes with the SARS-CoV. That means the previous pandemic, not the pandemic, it's previous epidemic of coronavirus, experimentally validated epitopes. That means those epitopes which are mainly validated in the experiment that they can generate the immune response. So we took only those epitopes which are experimentally validated in the SARS-CoV as well as in the SARS-CoV-2. They can generate both humoral as well as cell-mediated immunity. It does not have any mutation. They have very low risk to generate the antigenicity, toxicity as well as the autoimmune reactions. So this is mainly based on the computational prediction. So this is the main power of the computational vaccinology, or we can say that is in reverse vaccinology. And there is a new terms also, we can say that this is an immunoinformatics approach. That means we are using some kind of bioinformatics approach in the immunogenomics field to get the particular pathogenic sequences or the virulent sequences from the different viruses or the pathogens. And also we can predict which part of the viral sequences can bind with human immune system. Is called generally we call it major histocompatibility complex. And we can also, using this computational method, we can also calculate their binding affinity, how strong they can bind with their human system. Okay, so it is mainly based on a computational study. So what is the main importance of this computational study? Because suppose if there is a kind of pandemic or the epidemic situation again reappear, so then if we are going to search a vaccine candidate based on the experiment, there is then it can take several years to get which part of the vaccine or which part of the pathogen could be a vaccine candidate. So it can take say about 2 to 3 years if we are going to the conventional experimental method. So now the computational biology is a new field, and these approaches, different approaches of this computational biology could be used in this field to understand from the genomes, from the pathogens to which part of the pathogen's protein could be a vaccine target, and which this part can generate the immune response in the human or not. So it is a very interesting and important field, and these things could be changed the world, because if we know the computational biology, and if we develop the computational biology, then we can detect and predict many more things. Okay.
Right. So what you're saying is most of your efforts is based on prediction models, computational
Yes.
prediction models, right? So
Computational prediction.
in the real world, we have seen the coronavirus virus to mutate at a pace which has not really been predictable. So how far do you think computational models are applicable? Because they are basically models. They cannot match up to mutational possibilities, because mutations can happen at with a very wide range.
Yes, exactly. So for this reason, we have to take all the coronavirus data from genome sequence data or entire data from the databases from the last 10 to 12 years. Then you can check which part are mutated, which part are the conserved in those present SARS-CoV-2. As we can see that if in 2002 outbreak there is a SARS-CoV outbreak, and in 2019 there is a SARS-CoV-2, near about 17 years. But 85, if we compare the genome sequences between these two viruses, we can see that there is an 85% sequence similarity. 85%, only 15% is mutated. So from this, the main part of the SARS-CoV-2 which are evolved is the S protein. S protein is the spike protein. As I say that these spike proteins are the important to attach the virus to the host cell with the human ACE2 receptor. It interacts with the human ACE2 receptor. So this, as we say that there is an 85% similarity. So we can understand in the last 17 years, if the virus mutated enough, still there is an 85% similarity. So from this region, we can identify from the proteins or the genomic level or the nucleotide sequence, we can identify which part exactly evolving less or which part are evolving faster. So that means we can predict their evolutionary rate of particular proteins or the part of the protein sequences. And yes, this for designing a vaccines, as I say that we have to consider the part which are less evolving. That means there is a chance to get the less mutations. So if we select a particular portions from the viral sequences which are rapidly evolving, so then what will happen? Then if we design a vaccine again using this viral protein, then after some time, if the viral mutated these portions, then it will be problem. What problem? Then these particular protein sequences, if evolved, then it can, if it is we use this in vaccine, then it can bind in our human system. So again, if the pathogen reappears into our system, then our immune system does not recognize, because we have the memory cells for memory for the particular epitopes, and if the virus has changed these sequences, then they can escape the immune systems. So this is the most important thing. We have to consider the less evolving part of the virus. And yes, we can, based on the evolutionary analysis or the phylogenetic study, we can identify the particular regions from the pathogens which are less evolving.
Right. So you're saying the answer to this puzzle is a combination of statistics, evolutionary study, and computational biology.
Computational biology, yes.
Right. Right. So one last question, Dr. Mukherjee. This comes to you on a very layman's perspective. If I ask you, as a scientist, as a researcher involved very closely with vaccine making efforts, how quickly do you think we are getting a vaccine? A very random timeline. Give us if you could predict.
Yes, as I said, there is 14 vaccines which passed the preclinical trials, and it is under phase 2 or phase 1 or phase 3 clinical trials, and specifically not the only 4 vaccine, as I know, that is under the phase 2 or 3 clinical trials. So if we get the result from, I think we hope we can get the result of phase 2 phase 3 clinical trials by the end of this year, especially for these Oxford vaccines, because they are using the trial in the Brazilian populations. You can different part of the world, but we have to wait for more 6 months, because we have to check if there is certain kind of side effects, or it can generate the long-term immune response, or it can only generate the short-term immune response. So I hope we have to wait for near about 10 to 12 months from now to get a vaccines, but still it's an possibility, but we can't say that exactly yes there will be a vaccine in the next year or by end of this year. But yet, as for this pandemic, lots of research group and company and their collaborations are working to develop a particular vaccines based on the different vaccines for this particular COVID-19. So we hope we can get the vaccine in near 10 to 12 months.
In 10 to 12 months, and that's still a very very fast.
Yes.
It's very much earlier.
Very hard. Because as we can see that in Ebola, it's more dangerous than coronavirus. The outbreak was in West Africa in few years ago, but it took near about 15 years to get the licensed
Exactly.
vaccine for the Ebola, and it sanctioned in the last year, I think in 2019.
Exactly. Exactly.
Yes.
Right. So the next question that we have is, we have seen global statistics have told us that in the outbreak was particularly difficult in very densely populated and high populations countries of India, USA, countries like Italy, most European countries. But it was not as widespread in the Middle Eastern countries. Statistically, the infection rates were lower than India. Say, for example, India, the infection the death rates were lower than India. So, what steps, since you staying in Israel and researching over there, so what steps did the Middle Eastern countries take in particular that were very different from India, that led to the prevention of the spread of COVID-19?
Yes. So in Israel, I think few last month there is several decreases in the cases, but at present the cases are increasing exponentially. That means per day is near about 2000 2500 cases. So yes, when in Israel, uh, the peoples are generally at this in the last months or before that, there is a complete lockdown and everything. But when the lockdowns uh, rule, generally when the everything is started as usual, then again the infections reappears. So we can stop it. So generally it can't vary between uh, exactly the severity can depend between based on the region to region, but the infections rate or the
There's
infections
no predicting
progress
it.
yes,
Okay.
exactly.
Okay, right. So, Dr. Mukherjee, thank you very much for joining us today. And I hope
Thank you.
this your lecture has been very very enlightening, and I hope all our viewers have gained something from it. Thank you, viewers, for the questions that you have put up and engaging with us today. We will request all of you to subscribe to our channel if you want to go ahead and you want to see further of these coming up interactions with regarding issues which will be very very close related to current global situations and subsequent things. Dr. Mukherjee, thank you very much for joining us,
Thank
and
you. Thank you for inviting me.
we'll take your leave.
Okay. Yes. Yes. Thank you. Bye.
Bye.